What are indicators? Suppose you are in a laboratory and I give you two test tubes, one has an acid in it and the other has a base, and ask you to identify them. Now, how will you do that? You cannot taste or touch them as the acids and bases present in laboratories are very corrosive in nature. They can even burn your mouth or skin. So what will you do? The best and easiest way to identify the difference is by using an indicator. An indicator is a substance that changes its color when it comes in contact with an acid or a base. The two most common types of indicators are natural and synthetic indicators. Let's discuss natural indicators first. A natural indicator is a type of indicator that can be found naturally and can determine whether the substance is an acidic substance or a basic substance. One of the most common examples of natural indicators is litmus. Litmus is a purple dye that is extracted from plants called lichens. The natural color of litmus is purple. Now, this purple color dye is mixed with water and this becomes the litmus solution, which is obviously purple in color. What will happen if we add acid like hydrochloric acid to this solution? Well, the solution will turn red. But on the other hand, if we add a base, like soap solution to this solution, it will turn blue. So, to summarize, litmus turns red when in contact with acids and blue with bases. But the litmus is most commonly used as an indicator in the form of litmus papers. We have three types of litmus paper. One is blue litmus paper, other is red litmus, and lastly the purple litmus paper. These litmus papers are made by soaking filter papers with red, purple, and blue solutions mentioned before. Now coming back to the question we asked you in the beginning of the video. How will you identify which test tube has an acid and which one has a base? Yes, we will use the litmus papers to do so. So let's discuss it in detail. In the first test tube we will add a red litmus paper. We will see that there is no change and the color of the litmus paper remains red. Then we will put a blue litmus paper in the same test tube and we will notice that the color will change to red. Similar thing will happen with the purple litmus paper. It will turn red. The conclusion we can make is that the first test tube consists of acid. We will follow the similar process with the second test tube. The red and purple litmus paper will turn blue and the blue litmus paper will not change its color, indicating that the second test tube consists of a base. So, always remember, be it any colored litmus, acid changes it to red and base changes the litmus to blue. Now are you wondering how the color of litmus papers will change in a neutral solution which is neither acidic nor basic? Let's try putting the different litmus paper in a neutral solution and see what happens. The color of all the papers will remain the same. That means there is no change in the color of litmus paper due to the neutral solution. The next natural indicator that we are going to discuss is a common household item. It is turmeric. Yes, turmeric, which is so commonly used in Indian households, especially for cooking. Now we all know that turmeric is yellow in color, but have you observed one thing, that when you wash a stain of curry on a cloth with soap, it turns reddish brown? That is because turmeric is working like an indicator here. Soap is basic in nature. So, as soon as it comes in contact with turmeric, turmeric changes to reddish brown. But what happens to turmeric in acid? Can you think of an acid which is used in cooking that comes in contact with turmeric? Well, you may have observed that when we use turmeric in food and we squeeze lemon in it, which is acidic in nature, the color of turmeric remains the same. That means turmeric in acidic medium remains yellow, whereas in basic medium it changes to reddish brown. There is one more natural indicator, which is red cabbage, and it looks a little purple. The extract of red cabbage is used as an indicator. In acid the extract turns red, whereas in base the extract turns bluish-green. Even petals of some flowers like hydrangea, petunia, and geranium can be used as indicators. For example, if we take hydrangea, the flowers of hydrangea will appear blue if the soil from which they grow is acidic, and they will appear pink if the soil from which they grow is basic. So, the petals of the flowers of these plants can be used as indicators. So, until now we have discussed various examples of natural indicators. To reiterate, we have discussed litmus, turmeric, red cabbage, and petals of flowers like hydrangea, petunia, and geranium. Now, let us move on to synthetic indicators. Synthetic indicators are those which are man-made. The first one we are going to discuss is methyl orange. The name itself tells that it is orange in color. When acid is added in methyl orange solution, it turns red, whereas in base it turns yellow. The second synthetic indicator that we will be discussing is phenolphthalein. Phenolphthalein is colorless. In acid it remains colorless, whereas in a base it turns pink. If it is a very strong base, it may also turn purple. So, the two colors of phenolphthalein are colorless and pink. Now we have discussed ample examples of natural and synthetic indicators. But wait, there is one more interesting way of testing whether a substance is acidic or basic. Let's discuss it with an example. Let's say we have two beakers. Beaker A and Beaker B have acid and basic solutions, but we don't know which beaker has what. So, we add onion slices in both the beakers. You heard it right. We are going to add onion slices in both the beakers. What will happen? Are you waiting for the colors of onions to change? Well, let's check beaker A. It will have the smell of onions. Nothing new here. Now let's check beaker B. The smell of onions will vanish. This indicates that beaker A has acid in it, whereas beaker B has base in it. Well, such substances like onions which change their smell in acidic and basic medium are called olfactory indicators. Actually, olfactory nerve is responsible for the sense of smell. So, such indicators which change their odor in acidic and basic medium are called olfactory indicators. So, an onion is an olfactory indicator which changes its smell in base. We can try the same activity with vanilla essence. We put 2 drops of vanilla essence in beaker A which has acid and beaker B which has a base. In beaker A the smell still remains, whereas in beaker B the smell of vanilla essence has vanished. That means beaker B is a base. So, always remember that in acids the smell of olfactory indicator remains as it is, whereas in bases the odor goes off. This thing can also be repeated with clove oil. So in this video we have discussed what are indicators and the 3 types of indicators: natural, synthetic and olfactory indicators. Now indicators are an important topic for class 10 board exams. Let's discuss a previous year question from indicators. The question from 2019 board exam is, how is the presence of an acid tested with the strip red litmus paper? This question is a bit tricky because an acid will have no effect on the red litmus paper. Similarly, a neutral solution does not change the color of red litmus paper. So how can we differentiate between an acid and a neutral solution with the help of a red litmus paper? So firstly we will dip the red litmus paper into a basic solution. The basic solution will turn the red litmus paper blue. Then we will dip this blue litmus paper into the unknown solution. If the solution turns the blue part of the red litmus red, that means the solution is acidic, otherwise it is neutral or basic. Now it's time to practice more questions related to this topic. EduRef provides practice tests for all the topics of science along with chapter notes, videos, NCERT solutions, PYQs, and so much more. Hope you have enjoyed this video. Thank you.